All right, so hello everyone, and thank you for joining us today. My name is Chloe Halfhide, and I will be your host for this Precisionary Instruments webinar today. We're excited to welcome Zachary Saracino, a Postgraduate Associate at Yale School of Medicine. Zach works in the Chioccioli Lab in the Department of Comparative Medicine and Genetics, where his research focuses on regenerative medicine and stem cell dynamics during lung injury and repair. Today's talk is titled Cellular and Molecular Dynamics of Lung Injury and Repair. Zach will be sharing how precision-cut lung slices, or PCLS, and Lung Explant Imaging System, LEIS, can be used to study alveolar stem cell behavior and tissue regeneration over time. As a quick reminder, please feel free to submit any questions in the Q&A box during the talk, and we'll address them at the end of the presentation. And with that, I'll hand it over to what I'll do is I will mute myself and go off camera, and then I will have the presentation on my end. So Zach will be telling me to go to the next slide as we're going through this presentation. So I will mute.
Great, I think I can see that. You actually don't need to do presenter mode if it makes it, yeah, that's perfect. Okay, awesome. All right. Hi, everyone. Thank you for the kind introduction, Chloe, and for the opportunity to present today. Before we start, if anyone has any questions at all, please feel free to stop me in the middle of my presentation. I'm more than happy to answer them there. And of course, I'm more than happy to answer any questions at the end. So I'm very excited to be giving this talk for Precisionary today. Our lab started a little over a year ago. And today I'd like to give everyone an overview of some of what we do, the ongoing work, future directions, and the big questions that are driving us. And so we're interested in the mechanisms of lung injury and repair, and specifically how such a delicate and vital part of the body maintains its function under stress. And so our approach combines live imaging and genomic tools because to truly understand this system, we need to see what cells do and to observe their behavior in real time, not just identify what they look like. And so that's this kind of story that we're trying to tell in biology. Next slide, please. Thanks. And so these are sort of the main points that I would like to highlight in this talk today. The first being that PCLS, or precision-cut lung slices, are a great model for studying dynamic cell behavior in the lung. And second, that alveolar stem cell migration between individual functional units or cells is required for proper tissue repair and regeneration. And last, but certainly not least, that the Lung Explant Imaging System, or LEIS, that we have developed and optimized has helped us and will continue to help us study the spatiotemporal dynamics of AT2 cells following injury in greater detail and over an extended period of time. Next slide, please. But before we get started, why is studying the mammalian respiratory apparatus important? As I'm sure many of you have a relative or know a friend that is or has been impacted by some form of lung disease, lung disease is a huge burden, with 3 of the 6 causes of death worldwide being caused by lung disease, and the lung adenocarcinoma being the leading cause of death in cancer. And so here are a few of the many different lung diseases. You have some more acute diseases such as COVID-19, and then you have diseases like asthma, and of course the more chronic and maybe debilitating diseases such as COPD, cystic fibrosis, and idiopathic pulmonary fibrosis, which is what we have a model for in our lab. Next slide, please. And so the principal function of the mammalian respiratory apparatus is to maintain mammalian physiology through gas exchange. And so we focus most of our research on the site of gas exchange, which is the alveolus depicted here. And few fun facts about the alveolus is that we actually have 350 million approximately alveoli in each of our lungs. And it's the largest environmental epithelial interface with a surface area totaling about 70 meters squared. And just to give you an idea of how big that is, that's about the size of a racquetball court. And with 5 to 7 liters a minute of air being inhaled, you can imagine that our lungs are continuously being exposed to toxins and pathogens. And so the lungs have this finely developed system to maintain homeostasis. And so if we zoom into one of these alveoli, you have a bunch of different cells. Of course, you have your distal airway cells, bronchoalveolar stem cells, and then we have the cells that I focus most of my research on, which are alveolar type 1 cells, or AT1 cells. These cover approximately 95% of the surface area of an alveolus. And in terms of their structure, as you can see there, they're kind of flat and take up a large surface area. And underneath a microscope, they actually create this really cool web-like pattern that you'll see later on. And then their primary role is gas exchange, whereas their counterparts, alveolar type 2 cells, take up only 5% of the surface area of an alveolus. And they are much smaller compared to the AT1 cells and more cuboidal in shape. Now, they have two primary functions, one of which is to secrete surfactant, which helps maintain the surface tension of the lungs. And then the other is more of a stem-like function, which we'll talk about very shortly. And then, of course, you have your immune cells, such as alveolar macrophages and the like. Next slide, please. And so, as we just mentioned, AT2 cells can also act as stem cells, replacing themselves and AT1 cells. So under normal conditions and with homeostasis, there's, let's say you have an inflammatory response. Well, that leads to the proliferation of fibroblasts and myofibroblasts, which is eventually resolved through apoptosis. And if you can click next. And in contrast, in severe and chronic lung injury, the normal repair process is derailed or hijacked. And so AT2 cells don't repair properly in this case. There is persistent inflammation, and the extracellular matrix and myofibroblast accumulation does not resolve. And so all this, as you can imagine, leads to impaired gas exchange and reduced lung capacity. Next slide. And so what happens when AT2s are activated, undergo remodeling and repair? Well, at first, the AT2 cell sort of transitions into this intermediate progenitor cell, or IPC cell state that you can see there in the middle. And then from there, two different paths can be taken. More AT2 cells can be replicated if the body is in need of AT2 cells, or in the case of an injury, they can also differentiate into alveolar type 1 cells. And so we know this happens, but we want to know when, where, and how does this happen. Next slide, please. And so definitely AT2s are key players in this process, and that is why we decided to look at them when activated post-injury in greater detail. Next slide. And so that brings us to here. So to be able to follow the injury repair in real time, we've taken two approaches. One is lung tissue slice culturing, which I will be explaining on the next slides, as well as an in vivo lung imaging using a permanent lung window, which is what my colleague focuses on. So both of these allow us to revisit the same cell and region time and time again throughout the course of injury. And you can see what the lung window looks like down there with the cute little mouse who successfully recovered from his surgery. Next slide. And so I want to preface this by saying that this ex vivo portion would not be possible without this. And this is the Precisianary's compressitome, and our specific model is the VF5100Z. And so there's a few key features that I would like to mention with this instrument. On the interface on the right, you have an adjustable advance speed from 1 to 10, settings of 1 to 10, adjustable vibration frequency, also settings 1 to 10, and an adjustable thickness, which can actually be adjusted from 1 all the way up to 999 microns. And it has been quality tested to cut below 10 microns. And with some tissue types, you can actually cut as low as 4, depending on that tissue. And so there's also start and stop buttons and forward and reverse that you hold down to fix the white plunger in place. And you can swap between a continuous cutting mode, where once you hit start, the blade will keep slicing until right before you reach the plunger itself, and a single cutting mode, where once you hit the start button, only one slice will be generated at a time. And then you have to hit the start button again. Next slide. And one more, next. And so these are just the specs. I encourage you to take a look at Precisionary's website if you're interested in this particular instrument. Would highly recommend, 10 out of 10. Next slide, please. Thank you. And so this is actually my typical setup before getting started with generating PCLS. And so just in case you're curious, if you have any questions about how we harvest our lungs, I'd be more than happy to send you our optimized protocol, or also answer any questions that you might have at the end. But for this particular presentation, I wanted to focus on generation of the actual PCLS. And so in this protocol, you start with obviously attaching one of the magnetic blades that's there to the blade holder underneath that black part that's furthest on the white platform. Next, I fill a 12-well glass bottom plate with 1.75 mLs of DMEM F12 1:1 media. And in that media mix, I also supplement with 1% penicillin streptomycin to minimize contamination, 1% ITSG, which is insulin transferrin selenium. And that is really not necessary unless, if you want to include it, it can help reduce cellular stress, but you don't have to. And then, of course, 0.1% FBS. And that I want to really stress the 0.1%. We've found from personal experience and also from the literature that if you go above that, it's actually toxic to lung epithelial cells. So then I carefully transfer the lung, which I just acquired from the mouse, and I filled with 1.5% weight by volume low melt agarose to a Petri dish with blunt forceps. And then I separate a lobe with another blade. And then after this, I transfer 1 to 2 drops of super glue on top of the plunger, spread it around, and transfer the lung to the center of that plunger. And then I wait until it adheres. So you then want to pull the plunger down until the tissue is level with the metal surface and fill it to the top with agarose and add a chilling block until that solidifies. Next slide. And so just before you start slicing, you insert the apparatus with the lung tissue into the buffer tray that you see here, press the forward button on the vibratome to connect the metal rod with the base of the plunger, and fill the tray about halfway up with the media mix. And finally, we do a speed and oscillation of 5 and a thickness of 300 microns typically. One thing about the thickness is that if you want to, you can always increase the thickness. It just gives you a little bit of a buffer in case, for example, some of the agarose melts off and your tissue comes off with that. You lose some cells, so it gives you a few extra cells. You might just have to change out the plunger more frequently, but that's okay. So once you have all the settings that you are set with, then you can go ahead and hit start. And this is what you should see. You can hit next. So the vibrating blade comes down and cuts the tissue off with the agarose, and that gets deposited in the bath that you see there. So once these PCLS start collecting in the bath, I collect them with these yellow plastic spatulas that you saw earlier on that slide, create an 8mm diameter punch with the biopsy puncher, and transfer one of these per well, removing any agarose in the tray in the process. And there you have your PCLS. So from here, you can store them in the incubator and use them for the next part of your experiment. Next slide, please. So why PCLS, right? Why did we decide on this for our system and for the experiments that I'm conducting? Well, PCLS is a great system because it turns out that it preserves lung tissue structure and cell-to-cell interactions quite well. It's basically sections of live lung tissue that can be cultured ex vivo for 5 to 7 days. And of course, it's a great model for lung diseases such as lung fibrosis, and it offers a high number of replicates, both because you can image multiple wells, of course, at a time, and multiple locations per well. So you can actually generate many field of views per PCLS. And in case you're curious, that's what human PCLS looks like at the bottom there with a human lung versus the mouse PCLS that we typically work with on the bottom left, and bottom right, sorry. Next slide. And so we took advantage of PCLS to observe live cells in the alveolar region. In some of our first experiments, this was one of the first ones that we did, involved observing the same cells live over 72 hours. And so you, of course, in this system here, have a lid from the plate. And underneath that, you have a metal weight that holds down everything beneath that you see there. And that includes an inlet opening system, which is like a plastic membrane where media can diffuse in and out towards the cells. And then you have a 300 micron spacer that surrounds your 8 millimeter diameter PCLS. And then, of course, you have the PCLS that fits nicely within that space and is weighed down and fixed in place. And of course, you have your typical incubator conditions that we used, which is 5% CO2, 37 degrees Celsius, and 90% humidity. And we, in this experiment, imaged with the Andor Dragonfly spinning disc confocal microscope. Next slide. And what we see, which is really surprising to me, is that a lot of AT2 cells become motile, covering quite a distance within and across different alveolar regions that you can see here in this video. And I just want to make a note, how do we get to this fluorescence, right, that you see there? So our typical workflow can be seen on the left. That's the typical timeline that we follow. It involves, we use a tamoxifen-based system. So we inject our mouse with 1 dose of tamoxifen a day for 4 consecutive days. Then we wait 2 weeks. And then at that 2-week time point, which we now call day 0, we injure the mouse with, in this case, bleomycin administered intratracheally, as well as the control PBS. We then wait 3 more days. And then once we reach day 3, we sacrifice the mouse, harvest the lung, and generate the PCLS. And then from that time point on, we can image as long as we need. Next slide. And so I'm not going to go through all these data because they are published, but here you can see one of the most exciting AT2 cells going through a pore of cone. And so in this experiment, we used bleomycin, which, in case you didn't know, is a chemotherapeutic agent to induce acute injury to the lungs and damage AT1 cells. And so you can see in the control here that there's essentially 0 displacement on the bottom plot. Whereas at 3 days post-injury of bleomycin, you can see significant increase in displacement, which suggests that something is happening to the AT2 cells here to make them motile, potentially responding to the site of injury. And so what's also really interesting, if you look at 10 DPI, there is no displacement, because at this point, the AT2 cells have migrated to where they need to be and are actively responding to the injury. Next slide, please. And so this led us to ask the question about the stem cell domain. Is it local or is it global? Because if a stem cell can expand its spatial domain and migrate to where they're needed, that would be really cool. Next slide. And so here in this in vivo video that you can see here, there is a stem cell migration between alveoli, which is really cool to see. Next. And here are the individual timeframes of the video, so you can see exactly when it squeezes through the pore and transverses the adjacent alveolus. So if you look at 23 hours, it has its first point of contact with that AT1 cell barrier. And then at 30 hours, or by 30 hours, there's basically a complete transversal of that AT2 cell to the adjacent alveolus. Next slide, please. And to our knowledge, this is the first video of an AT2 crossing an alveolus. Next slide. And so this I wanted to highlight this example first. In this example, you can see both in the video and in the surfaces on the right. By the way, we analyze our live imaging data through Ameris, which is what we used, the software that we use to generate the surfaces that you see on the right. You can see this high displacement and elongation of the motile AT2 cells. Next slide. And in this example, you can see high displacement as well with this large cup shape that starts to form. And you can see a decrease in, at the same time, this decrease in AT2 sphericity and an increase in surface area in both these videos. And so I really want to highlight here how dynamic the behavior of AT2 cells is in response to injury. And you can see that you, one, have an increase in displacement in response to injury, this large displacement of AT2 cells. 2, you have this increase in the surface area as the AT2 cell differentiates into, remember, those more flat web-like AT1 cells. And then 3, you also have, of course, as the AT2 cell becomes less cuboidal, you have this decrease in sphericity. Next slide. So this led us to ask the question, if AT2s are not able to move after injury, what happens to the regeneration of AT1 cells? And so we used bleo in this experiment, and it turns out that when you deplete ARP-C3 in floxed mice, which, by the way, is a protein involved in a complex that nucleates and helps polymerize actin, AT2 shape remains cuboidal, as it does not have the ability to migrate to the site of injury and differentiate into an AT1 cell, as indicated by the decrease in HOP-X and tomato-positive cells compared to the unfloxed mice, which you can see in the plot on the bottom right. Next slide, please. So together, these results reveal that there's this unique stem cell migration between individual functional units and cells that is an important driver of tissue regeneration. And so these findings open up a new avenue for us, investigating the mechanisms that regulate stem cell migration and how these in turn are linked to tissue repair. Next slide. And so to do this, we're taking 2 approaches. The in and ex vivo approach that I mentioned earlier to follow individual cell transitions in real time before, after, and during injury. And of course, a multi-omics approach to understand the genetic mechanisms that drive the transitions. Next slide, please. And so that led us to the infamous Lung Explant Imaging System, or LEIS, that we developed and optimized, which allows us to manipulate conditions while observing the same cells live. And so as you can see in the schematic on the top, or actually the top row of images there, you have the schematic of the LEIS as well as the inner workings. And then you also have the typical setup that we have in both the incubator and the stage top incubator at the microscope. And so the setup is similar to before within the plate, where in my case, I use a 12-well plate. But you have an insert on top with an inlet and an outlet connected to tubing that goes through a peristaltic pump. And you can control this pump with a controller to indicate what time you would like to exchange your media. So for example, we do every 24 hours. At the 24-hour time point, the pump will transfer the waste media out of the well through the tubing to the waste bottle that you see there. And then the fresh media will be pumped from the feed bottle back into the well. And of course, you can fit this within the stage top incubator at the microscope, which is great because we want to do live imaging and we don't want the microscope, the feed, or the incubator conditions to interfere with each other. And so you might say, that's great and all, right? But how do we know it works? So we tested the viability of this system with 2 different assays. The first being an MTT assay. So the way an MTT assay works, in case you're not familiar, it's not like super duper common, but you treat the cells with MTT, this compound that live cells will take up, and their mitochondria have enzymes that will convert this compound into these purple formazine crystals that form on the outside of the cells. Now these crystals you resolubilize with DMSO, and once you resolubilize, you have this purple solution. So the theory is that the more alive the cells are, the more MTT they will convert to those purple formazine crystals, and the more intense purple color that you will get and the higher absorbance. And so you can actually see the results of these on the plot on the bottom left. You have here the resulting absorbances measured by the plate reader at 570 nanometers after 5 different time points for both PCLS with manually and automatically exchanged media. And of course, in addition to negative controls that we treated with 70% methanol. And what you see, not only do you see that there's no significant difference between the absorbances of cells that have their media automatically exchanged versus manually exchanged, but you actually see slightly higher absorbances in the LEIS. Now, nothing too significant, but it's still good news. And so the other assay that we performed was a caspase-3 assay. And in this assay, you might be a little more familiar with this. You treat the cells with this caspase substrate that is bound to a fluorogenic dye, and that readily crosses all plasma membranes of cells. You don't need to fix them, which is really nice. And the theory is that if a cell is dying, it's obviously undergoing apoptosis and expressing high levels of caspase 3 and 7 that will cleave that substrate, causing the dye to fluoresce green. And so dying cells will be positive for both DAPI, which I stain for, as well as the green caspase. And so when that happens, you know that you have a dying cell, and now when you have a live cell, it would just be positive for DAPI. And so the results are also very promising here. Very similar, going along the lines with the MTT results. There's no significant differences in the viabilities of cells that have their media automatically exchanged by the LEIS versus manually exchanged by hand. And so this is great because in the case of bleomycin injury, the peak is at 14 days and it's resolved by 28 days. So this allows us to study the spatiotemporal dynamics of these AT2 cells over these long periods of time without the cells dying or having to generate PCLS before, during, or after the injury peak. Next slide, please. And so I'm going to slightly pivot away from bleomycin, and I want to let you guys know, so we've used, obviously, the LEIS to aid with the study of bleomycin as an injury model and its effect on AT2 cells and AT1 cells. But we've also been more recently exploring this new injury model, which is the BHT model, which stands for butylated hydroxytoluene. And so this is a compound that's used as an antioxidant and preservative in many products and produces lung inflammation and selectively ablates AT1 cells via necrosis. And you give the mouse a single IP injection. It's really simple. It's also really cheap. And that concentration that we use is 400 milligrams per kilogram of mouse body weight. And so in this video that I'm about to show you, we also imaged at a 20x magnification, started at day 3 post-injury. And in case you're not familiar with the mouse line that we're using here, once again, it's a tamoxifen-based system. So once you inject tamoxifen, these mice are going to be expressing SFTPC, which is going to be represented by TD tomato in red, as well as HOPX EGFP, which will be represented by green. And once again, the typical setup is applied here, where we have a 12-well plate, 300 micron thick PCLS that are also 8 millimeters in diameter. We have 6 wells are control samples and 6 are BHT in a media exchange every 24 hours. So go ahead and hit next. So watch carefully here. I want to point out that around 3.5 days, the AT2s transition to AT1s, and just about right after those AT1s die, which is very fascinating. And how do we know this, right? How do we know this exactly? Well, if you go next slide, the AT2s begin to express high levels of GFP, in addition to all the other changes that we've seen in the previous slides. So if you hit next, take a look at the video on the left. I'm turning the channels on and off in this video to show you how AT2 cells are beginning to express GFP, which supports their transition to AT1 cells, along with the increase in surface area and decrease in sphericity that you see there. And if you hit next one more time, if we take a look at the video on the right, look at the AT1 cell in the middle that dies around frame 2 of the video, and around 7 to 7.5 frames, the AT2 is highly expressing GFP, which you can also see by the co-localization of both fluorophores, right? The red and the green color overlapping produce this really vibrant orangish like yellow color. Next slide. And you can hit next twice. Thank you. So, one more time, sorry. Thanks. And so research from Columbia, so this led us to explore in more depth the surroundings of these AT2 cells, right? And we found that in a study from Columbia that stromal cells, which make up connective tissues that support organs, actually transfer their mitochondria to mouse lung cells with acute inflammation. And they do this in one of 3 different ways. If you take a look at the diagram on the left, you can see that one is through tunneling nanotubules, which basically are a direct connection from the donor and the recipient cell. And the mitochondrion gets shuttled over from the donor to the recipient cell, where it then becomes a part of that cell's cytoplasm. You also have through extracellular vesicles. So, of course, right, the mitochondria will bud and sort of fuse with that plasma membrane and break off into a vesicle that then gets transported through the extracellular matrix and then fuses with the recipient plasma membrane and then into the cytoplasm. And then finally, what I thought was super interesting is that some mitochondria actually get transported in this free-floating mechanism, where they basically come out of the donor cell and are naked, if you will, in the extracellular matrix, and then get engulfed by the recipient cell and become a part of their cytoplasm. And so back to the study, researchers from Columbia actually found that cells with loaned organelles had higher concentrations of cellular ATP, which they actually ended up being distributed to nearby cells that did not receive new mitochondria. And what they found was that diseased lungs that were loaned mitochondria showed much greater signs of recovery. Next slide, please. So this led us to ask this question. Do AT2s swap their mitochondria with other organelles? And so before I play, before Chloe plays the video in the slide, I want to just preface this by saying we used a different mouse line, and this is the ROSA kaleidoscope line. And so in this this line here, you have the organelles that are able to be visualized. And what's really cool about this is you have, sorry, let me go to the next slide here. You have these the mitochondria that are represented in red and are expressing MK2. You also have the alpha tubulin that is expressing GFP and represented in green. And then you also have the lysosomes being represented by BFP in blue. And so, of course, we kept the same concentration of 400 milligrams per kilogram, and we used 20x magnification and started imaging at 2 days post-injury. So still to capture that 3.5 injury peak, day injury peak. And so you can go ahead and play the video. Oh, actually, sorry, hit next twice. It's okay, I think they're out of sync, the timer and the, that's okay though. So a few things I wanted to make note of here is that at the beginning, you see a high level of MK2 expression in the AT2 cells, maybe because there is a great amount of mitochondrial stress on the AT2 cells to produce ATP for replication, for movement, for differentiation, et cetera. And then over here, you can see this great extension of alpha tubulin in AT2 cells, which enables movement and attachment to other differentiating AT2 cells and might, right, who knows, enable transfer of mitochondria via the actin cytoskeleton, for example. Next slide, please. So obviously, the data that I just showed you in the past few slides is preliminary. So there's a lot of questions that are unanswered that I want to explore more and to hopefully be able to answer them in the future. And the first next step that I want to take is looking at that first mouse line that I showed you guys. And I want to repeat experiments to confirm that the AT2s do in fact become new AT1s over the course of about 3.5 days. And of course, I also want to perform a transcriptomics. I want to do a transcriptomics analysis based on single cell RNA-seq. And I'd like to obviously see what mechanisms are responsible for this AT2 cell behavior. I find it very fascinating. Like, for example, is it a mechanosensory response? Do AT2s sense a breach in the air-blood barrier in the lungs when AT1 cells die? Or is it a signaling pathways mechanism? Do AT1s release certain chemicals that AT2s respond to? Or, of course, is it both, right? And so I also want to make note that with the way that I would confirm this, in addition to the transcriptomics analysis, is generating more of the displacement, surface area, sphericity, cell number versus time plots of the AT1 and AT2 cells. And so then the second part of this is obviously looking more in depth of that ROSA kaleidoscope line. And I want to repeat experiments at a higher magnification. Approximately our 40x objective would be better, I think, to obviously observe the organelles in greater detail. And then also answer this question that we've been pondering. Is there a mitochondria transfer between cells in response to injury? And what drives this? Next slide, please. And so last but certainly not least, I'd like to thank everyone in my lab. So we are the Chioccioli Lab, led by none other than Maurizio Chioccioli. And I also have two amazing lab mates, Jameson Lead, who is a postdoc, as well as Lele, who is our new ARS. And I'd like to thank everyone past and present who has helped contribute to the projects I'm working on, and thank all of you for being such an amazing audience. And with that, I'll open it up to any questions.
Great. I just wanted to say a quick hello, Zach. I'm Abby. I'm Chief Scientific Officer of Precisionary. I had a conflict earlier, but my colleague Chloe took over, and I'm glad I was able to catch the majority of your talk. Actually, I had a question. When you had sectioned human PCLS and made the course, did you do that from like an entire lobe? How did you, because one of the challenges that we have experienced is being able to infuse agarose solution into a human lung sample, getting cores, and then making good sections. What are your tips and tricks for that?
So that's a great question. I wish I can comment more on that. I have not personally worked with human samples yet, but this is one that we're looking to get more of down the road. I would imagine from what I've seen, I actually watched a previous Precisionary webinar where I believe the presenter, he said that he worked with one lobe at a time, and they would punch the cores directly underneath the hood, directly from that lobe. And I think he inflated it, if I remember correctly, it was inflating with the agarose was right before he punched those.
Right. You had to inflate it first and then chill it so it solidifies.
Yes.
And what you want to do is kind of inflate the alveolar sacs before you section. Otherwise, it kind of collapse. Right.
Exactly.
I think the challenge is if you can't find a bronchial or a main bronchus to do the infusion, then how do you go ahead and make cores? Okay.
Oh yeah, that's a really good question, and I'm sure that I will be circling back to you and also my collaborators more when we start, hopefully, fingers crossed, working with human sections soon.
Excellent. Excellent. So I want to be able to open up the floor here. We have quite a few attendees to see if any of you would like to just unmute and or show your video and ask any questions. And if we don't have anybody, don't worry. With that, then often we get questions as a follow-up, and I'll be able to be happy to connect you with Dr. Saracino here. With that, I want to thank you so much, and also my colleague Chloe for hosting the beginning, and we hope to stay in touch.
Oh, sorry, really quick. I appreciate the title, Abby, but I'm not a doctor. I just want to throw that out there. Yeah.
No problem. Great.
Nice meeting you.
Thank you very much, everybody, for attending.
Yes, thank you so much.